15th International Dermatology, Cosmetology and Allergic Summit. I am Dr. Emenuga Veronica Ngozi, a lecturer with the University of Nigeria, presenting a paper titled Zoonotic Dermatophytosis, a Public Health Concern. Dermatophytosis are fungal infections of keratin on the skin, hair and nails. They are caused by fungal molds that require keratin for their nutrition. Zoonotic dermatophytosis are fungal infections of livestock and pets, but can be transmitted from animal to man. They are caused by fungi belonging to the genera Trichophytum, Microsporum, and Epidermophytum species. Humans are susceptible to these organisms. Transmission is from person to person, animal to person, soil to animal, and rarely soil to person. Zoonotic dermatophytosis is an important occupational mycozoonosis of dairymen, animal handlers, livestock farmers, pet owners, and veterinary officials. Some of the infections caused by this dermatophytosis are Tinea corporis, Tinea capitis, Tinea barber, and Tinea unguium. Zoonophilic dermatophytosis are found to have significant impact in public health and cosmetology, hence the aim of this research. The aim of this research is to identify the fungi causing this zoonotic dermatophytosis and also to suggest ways to prevent these infections. Materials and method. A total of 200 lesions were scraped, 100 from infected nomads from different animal ranches in Enugu State, Nigeria, and 100 from other artisans. Their lesions were scraped and taken to the laboratory for analysis. 10 cattle with fungal lesions were also scraped alongside, and their lesions were cultured, and the isolates were used for confirmation of zoonotic fungi. These were actually used as positive control. All the scrapings were inoculated onto Sabroidestrus agar containing 50 mg per liter of chloramphenicol, and the plates were incubated at 25 degrees centigrade for 5 days. After the 5th day, the colonial morphologies were studied for pigmentation, growth rate, and the texture of the growth. Microscopic examination using lactophenol cotton blue staining was used for the identification of the fungi. Slide culture techniques and some other biochemical tests were also done in accordance with Atanda et al. 2002. Statistical package version 20 was used to check for statistical significance of the results. Figure 1 here shows the fungal lesions on the goats. The figure 2 here also shows fungal lesions seen in some of the cow. Fig 3 here shows Tinea corporis lesion that were seen on the body of the nomad's wives, and Tinea unguium on the nails of the nomads. All these lesions were scraped and used for the analysis in this work. All these lesions were also cultured, and the causative organisms were used to analyze the result we got. Results. Of the 200 samples tested, 149 samples were positive for fungal growths. 8 fungal isolates were gotten. T. mentagrophytis, T. verrucosum, Candida albicans, Aspergillus species, Fusarium solani, Geotrichum candidum, Penicillin species, and Microsporum canis were isolated. Table 1 shows the percentage of occurrence of these isolates. All the 100 samples from the nomads yielded similar fungal growths as those ones seen on the cattle, while the fungi isolates that were isolated from the cattle were T. verrucosum, T. mentagrophytis, Aspergillus species, Geotrichum candidum, Penicillium species, and Microsporum canis. T. verrucosum and T. mentagrophytis and even Microsporum canis are dermatophytes, according to Meretti, 2013. Trichophyton species were the highest fungi isolated in this work, while Microsporum canis were the least in isolation. Of the 100 samples from the artisans, only 49 yielded fungal growths, and they were all not zoonotic fungi. There was statistical significant difference between the rate of fungal growth among the nomads and the artisans. Table 1 here shows the percentage of all the fungi isolates from the nomads and the artisans. From this table, we can see that T. mentagrophytis and T. verrucosum were highest in their occurrence amongst the nomads, while T. verrucosum was not even isolated among the artisans. So also, Microsporum canis was not isolated among the artisans. Fig 5 shows a slide culture of T. verrucosum, and Fig 6 shows the reverse side of the culture plates of T. mentagrophytis. Figure 6 shows a KOH mount of Microsporum canis. Discussion. The study revealed a high prevalence of T. verrucosum and T. mentagrophytis among the animals and the nomads. T. verrucosum and M. canis were isolated from the nomads and the cattle, but not from the artisan. This shows that the T. verrucosum and Microsporum canis are associated with the cattle, and the artisans do not have any contact with these cattle. The infection caused by these dermatophytes can also infect their family members. Hence, the nomad's wife were infected, and so it will spread to all the other public, causing public health problem. In conclusion, Trichophyton species and Microsporum species are part of the causal agents for zoonotic fungal infections, and they are communicable. Nomads are more exposed to zoonotic infections from animal and the animal farm area. Zoonotic dermatophytosis affects cosmetology and poses great health risks to the public because the infection is highly transmissible through fungal spores, which can settle on any immunocompromised skin at any time. Recommendation. We hereby recommend that there should be an adequately sanitized and clean environment for animal management, and proper sanitation should be maintained always. Nomads should protect themselves from getting infected, and they should also undergo medical checks regularly. Government should provide adequate medical facilities for animals and nomads in the animal ranches and farms. Thanks.